Many geneticists study genes. If you're going to study genes, they can exist in different organisms. This is why some geneticists study Drosophila, or some study yeast, or some study mice, some study humans. These other lessons which we learn from, say, Drosophila, mice, yeast, etc., often apply to humans. Many human genes were first isolated in other organisms, and we would understand human genetics far less if it wasn't for the study of the genetics of other organisms. Some geneticists study viruses, and there's a great deal which can be learned from viruses if you want to understand human genetics, or if you want to understand ecosystems in general. So the images here, a milliliter of water from sources throughout the world, and have simply incredible number of bacteria, which will be the topic of the next video, and also viruses. And so therefore, viruses are important in ecosystems. They're important for human health, and they can also be a source of understanding for just how genes work in general. So many geneticists study them. So this is going to be just a quick overview of some of the topics in viruses, some of the differences between them, and the idea of how geneticists can study. So first off, viruses are specific. There are viruses which infect cyanobacteria. There are viruses which affect protists. There are viruses which infect animals, but often only very specific species. So for example, the coronaviruses, which we'll get to in just a little bit, the majority of the viruses in the family Coronaviridae infect bats, but they can sometimes migrate to another species if the virus can then recognize the receptors on the surface. So there has to be a specific recognition. And this is why not only are viruses typically specific to one or several species, but also a cell type. So there are viruses that cause hepatitis in the liver but don't infect other tissues, or viruses which cause meningitis in the brain but don't infect other tissues, etc. Viruses may have to do different things, and so very often viruses that infect bacteria, for example, after they bind to their receptor, they have to literally inject their nucleic acid through the cell wall of the host. And so the first step in a viral infection is attaching to the cell surface and involving a specific interaction. And I'll get to more specific cases in humans in just a little bit. The nucleic acid in viruses is interesting because it can be double-stranded DNA, as in the cells of living things. It can be single-stranded DNA, but it can also be RNA in for example, the case of HIV or coronaviruses. It can either be single-stranded RNA or double-stranded RNA. Now, this is fascinating for many reasons. First off, if you're trying to combat the virus, it is useful to find differences between the virus and the human cells. So you don't want to, say, take a medication which will hurt a microbe that's causing an infection, but hurt yourself at the same time. So if you can find something that the virus is doing differently. And so some viruses using RNA, they then have to convert their RNA into DNA through a reverse transcriptase, doing the reverse of transcription, converting DNA into RNA. And so therefore a medication to treat the virus might then target that difference. Also, one of the interesting things about the RNA viruses is we believe that the precursors of cellular life lived in an RNA world where they used RNA for everything. And some viruses using RNA might actually hail from that time period. So if we're asking what did the original precursors of life look like, RNA virions, which are based in RNA, they may serve some answers. So the virus then typically gets its nucleic acid inside the cell. If the nucleic acid is RNA, it may have to be converted into DNA first. And then typically then what's called a lytic virus will then convert all of the cellular mechanics into making the cell into a factory to make more virus. So the host chromosome can be cut into pieces, and all of the ribosomes and enzymes can now be used to replicate the viral nucleic acid, and then to make proteins from the viral nucleic acid. So thus, a typical virus has only these two parts, a nucleic acid and a protein. And so once a virus infects the cell, it tries to convert the cell into a factory, which then makes more viral nucleic acid and more viral proteins, which can assemble into a capsid. The nucleic acid can then be incorporated into this capsid, and in a lytic virus, the host cell then bursts and releases all of these virions, where they can then start an infection cycle again. Now, viruses vary, and there are different ways that viruses can have a, quote, life cycle. And that's an awkward distinction, because viruses do vary. While we typically say that viruses aren't alive, it is true that there are some viruses which have larger and more complex genomes than some bacteria. So it's hard to make the argument that, you know, this thing isn't alive, even though it has more genes than this thing is. Some viruses have what's called a temperate life cycle, or a lysogenic life cycle, where their nucleic acid can then enter the host chromosome as in the form of what's called a prophage. And they're not going to cause the cell to burst, at least not right away. And they can sit dormant for a while. And now each time that the cell replicates, it's going to replicate the viral DNA along with it. The virus may at some point later initiate a lytic cycle. So perhaps if the cell is thriving, the virus may sit dormant, with the idea that the more the cell thrives, the more copies of the virus are being replicated. But if the host were to enter into hard times and be stressed, that the virus would then initiate a lytic cycle. So for example, HIV can stay dormant for years, up to 10 years. And this is why, you know, those who aren't showing symptoms can actually be spreading the virus. So those are just some different kinds of viruses, and I'll get to specific examples. It's common to use viruses in genetic studies, even if you're not interested in, say, health applications or ecological applications, but just to understand genes. If you ask why, well, let's look at fruit flies. We use them for genetic studies because they're small, they're easy to keep, their generation time is short, and so therefore you can do experiments with fruit flies that you could never do with, say, you know, dogs, or wait for human generations to pass, etc. Bacteria are even more so, where bacteria are even smaller, even cheaper to keep. And then viruses, this is even more the case. And so if you were studying genes, having something which is so small and so cheap is an advantage. Because if you're looking for rare events, if something happens very unusually, then you might have to count an incredible number of fruit flies in order to actually observe one. But if you're looking for, say, mutations, it's very often the case where you can start with, say, bacteria, because if you, say, start a bacterial culture in a shaker bath when you go home, when you leave work to go home, then overnight, it replicates so many that now you have millions and millions of bacteria to work with. And if there are very rare events, so if this mutation which occurred, let's say this is, you know, a new mutation for a bacteria which can now live on, this medium where before it couldn't. Let's say this occurs only once every 10 million times. Well, if you wanted to count 10 million fruit flies, that would take forever. But here now, here is a very rare event which is documented. So we have a dilution plate. We diluted it by a factor of 10 million. We saw one, let's say that was a mutant colony. Then you would be actually now measuring a rare event which occurs maybe once every 10 million times. Well, the same thing with viruses. Viruses vary. You can have mutant viruses, just like there are mutant fruit flies and mutant bacteria. And the viruses, one of the things that they can vary in is what hosts they can infect. And so a very commonly studied system is to use bacteriophage, which infects bacteria. And so E. coli can have different strands. There are E. coli K, E. coli B, etc. And so if the restrictive host, E. coli K, could infect, now be infected by the wild type virus, but not the mutant viruses. So if mutant virus can't infect this strain, but it can infect this strain, you could then study that. And you could then study, well, could there be a reversion mutation for the mutant strain to then become the wild type strain again, so that this strain, which can infect E. coli B, but not in E. coli K, could this now mutate so that it could infect E. coli K? You're not looking for a mutation, you're looking for a mutation at a specific nucleotide to go from here to there. This cytosine to this thymine. So what is the frequency that one specific nucleotide will undergo this type of mutation? Well, this is an extremely rare event. And if you wanted to, once again, study any other organism, you'd be counting incredible numbers of them. But because you can have so much virus in a milliliter, even these events, which might only occur, once per 100 million replications or once per billion replications, nevertheless, you could measure it and thus get data on this. So recombination within a single gene, that's a really rare event. But once again, if you wanted to study it, don't study fruit flies, don't study mice, study viruses. And so viruses are also then useful as tools as we attempt to understand just how genes work in general. Now, I'd like to just go through a couple of other variations. So if we were to consider, say, HIV, HIV is a virus that only infects certain cells. There has to be a specific receptor known as the CD4 receptor. So the virus has proteins which recognize this. And viruses can enter cells in different ways. In this case, the cell, after it has bound a receptor, can actually take in the virus through endocytosis. The virus takes advantage of a mechanism to bring things into the cell that the cell already has. HIV has RNA as its nucleic acid, so it must be converted into DNA first. And the DNA can then be incorporated into the host chromosome, where it can serve as a prophage. Now, when the viral particles are produced and leave the cell, they bud from the cell membrane. So they don't burst the cell all at once. They leave by budding, and thus they leave in a little membrane. So as they leave, they kind of look like a little human cell wrapped in human cell membrane. This is both good and bad. It helps them to evade detection by, say, antibodies, because you don't have the viral protein capsid entirely exposed, or it's wrapped in a human membrane, so it's easy to hide. But then as far as control, anything which causes the membrane to burst, like cold surfaces, disinfectants, etc., then would render the virus non-infectious, even though the virus was still there, because the membrane was not intact. It would be no longer capable of causing an infection. So there are viruses which have RNA as their nucleic acid. There are viruses which bud from the membrane and take some of the human cell membrane with them. And viruses, including HIV and herpes viruses, can hide inside the human genome. In fact, when we study the human genome, about 1% of human DNA originally wasn't human. It seems to have originated from endogenous retroviruses, viruses which inserted themselves into the genome. Now, if that happens in my cheek cells or nerve ganglion here, that obviously doesn't affect the next generation. Cheek cells don't make babies. But if the virions infect cells which are involved in gametogenesis, then you can now have viral DNA then become a part of the human genome. And once again, about 1% of our human DNA wasn't human. Now, just a few words on coronavirus. Coronavirus is a virus, and just as we refer to living things evolving over time, so too do viruses. New species of living things pop up, new viruses. And so in the coronaviruses, which primarily infect bats, sometimes a change occurs so that one is now able to infect another species if the receptors are similar enough. So these red spike proteins on coronavirus, they recognize a human protein known as the ACE2 receptor. So the ACE2 receptor, the angiotensin converting enzyme 2, this is the receptor for the virus. And then if we wanted to ask why viruses infect some species and not others, we would compare the receptor proteins. I'm just going to quickly overview some of the things I have on coronavirus, and then end with showing a playlist, which I have a number of additional videos that go into the topic in greater detail than this. So the ACE2 receptor, it's actually a protein which helps to lyse the viral spike protein. So it's an enzyme which actually breaks down proteins, but the virus is using that as a trigger to insert its genome into the cell. The genome of coronaviruses, they are RNA. They are single-stranded, so they must make themselves double-stranded, but because they're single-stranded and what's called positive sense, the minute it's inserted into a cell, it looks like an mRNA. So human ribosomes can start making viral coronavirus protein immediately because a single-stranded positive sense RNA molecule looks like an mRNA. All of the virus is in one single molecule, and this is different from say flu viruses, where there's different RNA molecules. The virus is segmented. Viruses often have, quote, a life cycle, if you will. So we can just get to this part. So here you can see that just as animals evolve and form new species, so too do viruses. So the coronavirus family has evolved and is continuing to evolve new members, some of which infect humans, such as the SARS coronavirus 1, the Middle East respiratory syndrome MERS virus, some coronaviruses which can cause the common cold, and then the coronavirus which causes SARS-2, the COVID-19 virus. And once again, it's RNA, and just to point out some of the finer points, viruses have a life cycle. So at the beginning of their life cycle, quote life cycle, they might want to replicate their nucleic acid, and then later they make the proteins of the capsid. So there's actually stages where they don't make all of their proteins at the same, or replicate all of their genes at the same time. And so in the coronaviruses, there are different sections. The earlier sections, these are involved in making more of the RNA molecules, among other things, copying it, proofreading it to limit mutations, etc. And then this part of the viral genome, it encodes many of the proteins which will form the capsid and help to package the virus. Coronaviruses belong to a group of viruses called nidovirales because of this nest, nido, which is the translation of nido, nest, because you have these proteins, the spike protein, the envelope protein, the membrane protein, the N protein, all coded in the same order at the end of the genome. And so what happens in the coronavirus infections, much more on this, is that the virus can start making its copies of this and proteins immediately because it looks like an mRNA. The proteins which are made help to replicate the virus into a negative sense strand, which then makes more positive sense strands, which can then be packaged into more virions. And so those last proteins include the spike proteins, the envelope proteins, the M proteins, which will attach the N proteins, and then some variable ones here. The virus then also makes what are called polyproteins out of these first two open reading frames, that one and the next one. So it makes this big protein that then gets cut into a lot of little pieces, and then each of these pieces has a job. So this makes a huge polyprotein, this one makes a huge polyprotein, and they all get cut into to pieces. And so inside an infected host cell, you have human ribosomes thinking that the viral RNA is mRNA, and so therefore it makes those polyproteins, which then take the viral RNA and copy it and make a lot more, so that now more infectious agents can be spread. And just what you're seeing here is I had said that the virus, if it's going to infect a cell, it needs a trigger. And the spike protein, the ACE2 receptor cutting the spike protein, that's the trigger. In addition to the low pH of this vesicle. So human cells, like in AIDS, they took this virus into the inside the cell after it bound the receptor, and then started to make this more of an acidic vesicle that could undergo digestion. But the virus actually wanted that, and is using the acidic pH and those enzymes there as the trigger in order then to get the viral nucleic acid inside the human cell. So once the RNA is is inside the human cell, the human ribosomes attach to it, and they start then to then make those polyproteins, which then causes more of the viral nucleic acid to be made. And then ultimately, you have all of these proteins which are being processed at the RER and other membrane systems like the ER to Golgi compartment here. And now that you have viral proteins in human cell membranes, now you have all of these viral these copies of the viral nucleic acid, then it then gets packaged inside virion. I have more on that elsewhere. I'll point you to that in just a second. But now that you have the virion with the viral proteins in human membrane with the viral nucleic acid inside, now it can then be released from the cell and bud from the surface. So here is that N protein here in blue attaching to the M protein. So the viral proteins inserted themselves in human membrane in these membrane compartments inside the cell. The viral nucleic acid was attached, and this then formed the virion that curved with all of those viral proteins now on the outside. And now it buds from the cell one at a time. So I've put together, and I will be continually adding to a playlist on coronavirus. Obviously, it's of tantamount priority to understand this. And so there are more videos here. But geneticists don't only study viruses because of the diseases that they cause. Once again, that just we can learn about genes in general by studying viruses. And then also, we are part virus. Our ancestors got viruses, and some of these viruses inserted their DNA into the cells of the germline. And so about 1% of our DNA didn't start off as being human. Somewhere in our ancestors, maybe some going back to early primate ancestors, some going back to early amniotes, that some of these viruses inserted themselves in the germline. And so if you go to different human chromosomes, you can recognize this originated as a virus. This is homologous to a virus. Now, obviously, we want to say bad. Oh, that's bad. It can cause mutation. And yes, that's true. For example, I don't want to get any more mutations. If I get more mutations, that might mean that, say, I develop skin cancer, so that would be bad. But then on the flip side, mutations obviously aren't all bad because humans are mutated apes. If apes never mutated, then apes would have stayed in their early generalized form. Mammals are mutated reptiles. Reptiles are mutated amphibians and the like. And so if viruses are a source of mutation, well, many of those may be bad. If our lineage had these retroviruses become part of our genome, we wouldn't be here without them. All right, that is part of what made us, human. Once again, mutations, we look at it, as being, this is this bad thing. We don't want change. But obviously, if, organisms didn't change over time, our species wouldn't be here. And so viruses are one of the agents of change. And there are plenty of endogenous retroviruses which have worked themselves into our genome and thus can change levels of gene expression, etc. So in general, viruses are important to study for geneticists for a number of reasons.